morning, students. So let us go for our next session in this first chapter, the living world. Fine. Hope you are fine and safe at home. So before we move to the topic, of course, we can go for this one review regarding this previous class. Fine. Okay. So the last class we discussed regarding these three domains of life, right? And this five kingdom classification. So three domains we said: domain archaea, bacteria, as well as eukarya, right? And this classification systems: five kingdom, six kingdom, as well as seven kingdom. Clear? And let us continue today's portion. Fine. So today we are supposed to discuss regarding two concepts. The one is concept of species, and the second one tools of taxonomy, right? Let us see the first one. Concept of species. So species are already we said in the last class. So species is a basic unit for classification, right? So one of this example even we have discussed in the last class regarding this classification of human being, right? There at the bottom, I mean at the last, at the basic, as it is given as species, right? And here if you scientists, they have given their own terms for species. Let us see the first one. So this term is given by the person John Ray in the year 1693 in his book Historia Generalis Plantarum. Historia Generalis Plantarum, right? Okay. Now let us see what is the term he has given. He said species is a group of morphologically similar organisms arising from a same or common ancestor. So of course if we say one species, it will be morphologically similar. Alright. Then it is arising from the common ancestor. This if it is one species, of course it may evolve or arise from the same ancestor. Previous animal is same. Understood? For example, Homo sapiens are one species. They are evolved from the Homo erectus. Understand? So that is a concept said by this John Ray. Fine. And he is Carolus Linnaeus. He has given the term for the species in his book Systema Naturae, right? And he said species as a basic unit of classification, of course. So with the species name only, we could identify the exact organism, right? That's why he said species is a basic unit of classification. Clear? And he is Charles Darwin, and he is one of the very famous and familiar evolutionist. So he has connected this word species with the evolution. Okay, and his book is Origin of Species. It is published in the year 1859. Okay, and his concept is evolutionary connection of species by the process of natural selection. And whatever species is there available in the world will be selected by the nature. That is a concept of Charles Darwin as per evolution. Okay, so what he is connecting this word species with the evolution. Fine. So this is the concept of species which is given by these three scientists, right? Let us go on the next topic. It is tools for the study of taxonomy. Okay, tools for the study of taxonomy. So what is taxonomy? Yeah, of course, this the science which is used to learn the classification of organisms are known as taxonomy, right? And what do you mean tools for study of taxonomy? What are the things is helping for the study of taxonomy? So what are the tools are helping to identify organism or identify one species? That's we can say as tools for study of taxonomy. Okay, here few tools is displayed. Let us see one by one. Fine. So the first one is the classical taxonomical tools. The second one, molecular taxonomical tools. The third one, automated species identification tools. Okay, and the next one, neo taxonomical tools. Then ethology of taxonomical tools, and the final one, e-taxonomic resources. Okay, so these are all the tools are going to help for the study of taxonomy. Okay, and let us see how each thing is helping for each. Fine. Among these, the first one is classical taxonomic tools. So classical in the sense, of course, we can say it's one of this traditional method. It is a classical taxonomical tool. So traditionally, what are this method is available to know about this taxonomy? Fine. Among this first one is taxonomical keys. The second one, museum. And the third one, zoological parks. The fourth one, marine parks. And the fifth one, printed taxonomical tools. Okay, let us see one by one. So the first one is taxonomical keys. Okay, so keys means actually, so whatever it is, if this information is given in a very short form, we could say this as keys. Okay, so here one of this figure I displayed also one of the method to identify one species. But here the information is given as just keys as a questions. But the answer is just a short form yes or no type question. That's all. Fine. For example, we wants to know about this gorilla, we could say. Okay. We can go for this question answers. We have the answer yes or no. That's all. Fine. So the first question is whether this animal has dry skin. Okay. Maybe our answer will be yes. Right. Okay. And the second question, whether this animal has hair. Of course, our answer will be yes. Then we will be moving closer to that animal, right? Okay. And the next question is whether it has opposable thumb. The thumb is whether it is in opposite direction than the fingers. Okay. Of course, our answer is yes. Okay. And the next question is whether it is bipedal. Means whether this animal is walking with the two legs is the question. So we are answering no. Then it is a uh, gorilla has four legs. But if it is two legs, we can say it leads to human being. Okay. This is one of the simple method for this. Example for taxonomical keys. Fine. Okay. And the next one is museum. Of course, everyone may about this museum, right? You may met this museum. So what will be there in the museum? Here in this biological museum, plants and the animals will be kept safely in a preserved form, right? Either it will be the extinct animal or living animal, but it will be preserved and kept safely in the museum, right? Okay, that is known as museum. So these museum also helping for the study of those any one species, right? So this is one of the zoological park. So zoological park, what do you mean? It is different from the museum. In the museum, the organisms are kept safely in a preserved form. But here, the organisms are left free, and it will be under the control of these human beings, right? That is known as the zoological parks. So with this behavior, with the behavior of these animals, also we can understand or we can study about this taxonomy. Clear? It's one of this marine park. So like zoological park, it is marine park. So here the fishes will be kept safely in the marine parks, and with this behavior, of course, with this morphological character, all those things, we can study this the species characters also. It is helping for taxonomical studies. Fine. And the last one is printed taxonomical tools. Here the information is given as a printed form. For example, like identification cards, or like as a descriptions, or as a manuals. It will be in a printed form. Okay, so this details will be given as a printed form, that is known as a printed taxonomical tools. So with these tools we could understand or analyze about these organisms. Understand? So these things we have discussed below this classical taxonomical tools. So what are all classical taxonomical tools? Taxonomical keys, museum, zoological park, marine park, as well as printed taxonomical tools. Okay, so these are all helping for the analysis of animals. Is it clear? Let us go for the next one. Molecular taxonomical tools. You know, advancement in the technologies is leading to the formation of molecular taxonomical tools. So here on the previous things we said, with the zoological park, with the marine parks, with the printed tools, we could understand or analyze these organisms. But here it is moving in a very basic level, even in a DNA level we can analyze these organisms, right? That method is known as a molecular taxonomical tools. So here also few method is given, that is DNA barcoding, DNA hybridization, DNA fingerprinting, or FLP means restriction, fragment, length, polymorphism, and PCR means polymerase chain reaction. So these are all this method is helping, or procedure is helping for this study of taxonomy. Understand? Let us see how it is helping for the taxonomical studies. The first one is DNA barcoding. Have you seen this barcodes? Of course, whenever we get the products, whenever we purchase this product, there will be the barcode, right? So when we go to the supermarket, this barcode scanner will be there. When they show this barcode scanner on this code, they will get all the details regarding the product. The barcode will be as parallel line and with a different width, right? With some sufficient gap. So each and everything has a information. So below that some number will be given. So each and everything regarding that product, whatever information is needed, everything is given as a code. Understand? That is known as a barcoding system. The same technique they are applying in this DNA also. Here, whatever the species we take, every organisms may consist of the cells consist of DNA. So DNA consists of DNA bases will be there, right? So the details regarding the particular DNA will be in the form given in the form of barcodes, like a product. So here the product details, everything will be given as a code. Here the DNA details will be given as a codes, right? So that is known as DNA barcoding, right? So when we go through these barcodes, of course we can able to collect the complete details regarding that particular species where from the DNA is collected. Understand? It is one of the new technology we can say. Fine. Okay. On DNA hybridization method. So DNA hybridization is nothing else. Have you heard this word hybrid? Yeah, hybrid means it's a combination of two different species. That is known as hybrid. Here also the same method. Here two DNA is a DNA A, DNA B, and DNA is a double helix structure. So it will separate each other. Both this DNA will separate each other in the second figure, and it will recombine. A will combine with the B and the B will combine with the A, right? So the new hybrid variety will be produced. So with this technology also they can analyze this about this species. Understand? DNA fingerprinting. So DNA fingerprinting is different than the DNA barcoding. Okay. So what is the difference here? The fingerprinting means actually the fingerprint, not the DNA fingerprinting. Usually the fingerprint is unique for each and every person. Am I right? That you know, right? The thumb or the fingerprint is unique. The same term they are applying for the DNA. Why could they apply the same term for the DNA? Because The DNA also unique for each and every person. So how it is unique, if we know this structure of this DNA, we can understand DNA consists of some bases. For example, adenine, guanine, thymine, cytosine. So it will be there in the DNA structure. Okay. So these bases will be repeated throughout the length of this DNA. But this repetition is not similar for each and every individual. It is unique for every individual. Understand? This repetition will be unique for every person. It will not be similar for everyone, right? So that's why they have using this term DNA fingerprinting. It is unique for each and every person. So with this technology also they can analyze this every species, right? RFLP means restriction, fragment, length, polymorphism. Actually, it is one of the technology. So by this method, the DNA will be fragmented. It will be cutted into different pieces. That's all. That is the word fragmentation. So every pieces or fragments may have this information. So as per that, the DNA studies will be taken, right? So this method also they are applying for the taxonomy. Okay. So the next one is PCR, means polymerase chain reaction. So this is also one of this current technology. So here many fragments or the length of this DNA will be produced, multiplied. For example, the first figure DNA is there, and the second one, DNA two strands will be separated, and the third step here, the separated strand will produce a new strand. So the two strand become as a two DNAs. Then the process will be repeated for so many times and the millions of copies of the same DNA will be produced. Actually, this is one of this technology. So this technology also they are applying for the taxonomical tools. Fine. Okay. So the last one is automated species identification tools. So it consists of cyber tools, means the information we could collect from the internet is known as cyber tools. So here the keys are given. For example, ALIS means automated leafhopper identification system. So if we go through this website, we could understand about this information regarding this leafhopper. We could collect the information regarding the leafhopper from the internet, right? And the next one, DAISY means digital automated identification system. Digital automated identification system means we can collect this information in the digital form from the system internet. The next one, ABIS, automated bee identification system, means we can collect the details regarding the bees. Next one, SPIDA, species identified automatically. For example, regarding spider, wasp and bees. Fine. And the last one, drawing, means honeybee wing identification. Honeybee wing identification. If you go through this website, we could understand regarding this details of this each varieties. Fine. Next one is neo-taxonomical tools. So here they are using these microscopes. So a lot of microscope is available, and this an advanced one is this electron microscope. So here even the nano level organisms also we can get the details with this microscope. So this microscope also helping for the taxonomical studies. Fine. That is known as neo taxonomical tools. Next one is ethology of taxonomical tools. Ethology means as per the behavior of these animals, this organisms will be analyzed. For example, behavior of these birds, sound of these birds, or bioluminescence of this animal. So bioluminescence means the animal from the animal's body the light will be emitted, right? So with these characters or behaviors, we can analyze or understand about these organisms, right? So this is the last one, right? E-taxonomic resources. Here, this informations we can collect as a databases from the system, from the computer, right? And one of this example we can say as INOTAXA, means integrated open taxonomical access. Okay, so first it is developed by Natural History Museum in London. So this informations we can collect as a databases from the computer, right? That is known as e-taxonomic resources. So these are all the tools is helping for the taxonomical studies. Fine. Hope you understood, and let us go for some assignments. So define species. So three species definitions we have learned that you can learn. Okay. Then second one, explain classical taxonomical tools. So classical method, traditional methods, what are the things we said? Fine. And how molecular taxonomical tools help in taxonomical study. So to molecular methods, what are the methods we said? That you have to write here. Fine. And the last one, explain automated species identification tools. That is the last one which we discussed now. Fine. Okay, so please go through this videos, watch these videos completely and collect these notes even from this videos also. Fine. Okay, be safe at home and pray well. Okay, we can meet in the next class. Thank you.